Companies must demonstrate during validation that the cleaning procedure routinely employed for a piece of equipment limits potential carryover to an acceptable level. The limits established must be calculated based on sound scientific rationale. This video provides practical guidance as to how those acceptance criteria can be calculated. It is important that companies evaluate all cases individually. There may be specific instances where the product mix in the equipment requires further consideration. The acceptance criteria preferably should be based on the acceptable daily exposure, ADE, or permitted daily exposure, PDE, calculations whenever this data is available. The ADE and PDE define limits at which a patient may be exposed every day for a lifetime with acceptable risks related to adverse health effects. Calculations of ADE and PDE of APIs and final intermediates are usually done with involvement of industrial hygienists and toxicologists, who review all available toxicology and clinical data to set the limits. The justification of the calculation should be documented. For API manufacture preceded by another API, when limited pharmacological or toxicological data is available, preliminary ADE and PDE with available data or TTC approach is recommended. In other cases where availability of pharmacological or toxicological data is limited, for example for chemicals, raw materials, starting materials, API intermediates cleaning limits. Based on the threshold of toxicological concern, TTC, LD50 and/or general cleaning limits may be calculated. In these cases, carcinogenic, genotoxic and potency effect of these structures should be evaluated by toxicologists. The acceptance criteria for equipment cleaning should be based on visually clean in dry conditions and an analytical limit. Acceptance criteria using health-based data. The maximum allowable carryover, MACO, should be based upon the health-based exposure limits, HBL, which can be an acceptable daily exposure, ADE, or permitted daily exposure, PDE, calculated when sufficient data is available. The principle of MACO calculation is that you calculate your acceptable carryover of your previous product, based upon the HBL, into your next material. As you see in showed equation, MACO is maximum allowable carryover, acceptable transferred amount from the previous product into your next material, and the unit is milligram. HBEL is health-based exposure limit, milligram per day, of the previous compound. MBS next is minimum batch size for the next materials, where MACO can end up, and the unit is milligram. TD next is maximum therapeutic daily dose for the next material, milligram per day. PF is purging factor reflects the ability of a process to reduce the level of the previous product in the downstream synthetic route of the next material. SF is safety factor reflects the effects from the interaction between previous product and next material. This factor should be applied in case of a risk for patient safety. Possible risk are for example contraindications, possible allergens, risk for children, previous products that should not be taken daily, next material which is only applied once. Assessed by a toxicologist. In case of no effects from the interaction between previous product and next material can be found, the default value is 1. The HBEL should be calculated as an acceptable daily exposure, ADE, or permitted daily exposure, PDE. They are effectively comparable with each other and represents an estimate of a daily exposure that is unlikely to cause an adverse effect if an individual is exposed, by any route, at or below this dose every day for a lifetime. They are determined to protect patients and are calculated by following formulas in milligram per day. ADE, acceptable daily exposure milligram per day. PDE, permitted daily exposure milligram per day. POD, point of departure. BW, is the weight of an average adult, e.g. 50 kg CFR EMA guideline. UFC, composite uncertainty factor, combination of factors which reflects the interindividual variability, interspecies differences, subchronic to chronic extrapolation, LOEL to NOEL extrapolation, database completeness. NOAEL, is no observed adverse effect level, mg per kg per day. NOEL, no observed effect level, mg per day. MF, modifying factor, a factor to address uncertainties not covered by the other factors. PK, pharmacokinetic adjustments. F1 to F5, adjustment factors to account for uncertainties. Refer to EMA Guidance 2 for further explanation. Point of departure. The point of departure is the dose level from which the HBEL is extrapolated. The point of departure can take many forms. It might originate from animal or human data, and the dose level can correspond to different effect levels. It is also dependent on the phase of development of the drug product at the moment of assessment. In later phase of drug development, more and more data become available and several PODs can be selected. In this case, the most relevant or conservative HBEL should be used. The most appropriate POD, however, should be carefully selected by expert judgment. In order to calculate an HBEL, the NOAEL or LOAEL should be available as POD. However, this is not always the case, certainly not for drugs in development. If there is no NOAEL or LOAEL available, LD50 can be used as POD. However, in this case a conservative approach is needed and therefore more uncertainty factors need to be applied. Other available data might also be used in order to define an HBEL, but this is based on expert judgment. If no data at all is available, the TTC principle according to Dolan et al. Should be applied. Drug products and APIs should have at least one or several NOAEL or LOAEL values available. Only very occasionally, for example in early drug development stages, no NOAEL or LOAEL might be available and LD50 values can be used, but only with very conservative uncertainty factors. It is, however, strongly advised to restrict the use of LD50 as POD in this case, as LD50 values are not reliable for predicting long-term effects. For intermediates where limited data may be available, HBEL determination guidance will be given by the toxicologist. For most solvents and detergents, HBELs are already determined and available in public databases, ACGIH, OSHA, MAK, NIOSH, etc. In general, the HBEL should be determined based on following hierarchy. 1. HBEL available, mostly for solvents and reagents use most stringent HBEL. 2. No HBEL available, but NOAEL or LOAEL available, calculate HBEL, as described, based on NOAEL or LOAEL as POD. 3. No HBEL available, no NOAEL or LOAEL available, use other available numerical data as POD to determine HBEL, like LD50 values. 4. No HBEL available, no other numerical toxicological data available, use other available data to determine HBEL, mutagenicity, carcinogenicity, CLP, etc., but this is based on expert judgment. 5. No data at all available, use default based on TTC or additional testing. This hierarchy should strictly be applied in setting the HBEL. The most reliable source of data available at that moment of assessment should be used to determine the HBEL. In cases where no other data is available and only LD50 data is available, the HBEL can be based upon LD50 data. Calculate NOEL according to the following equation and use the result for the establishment of HBEL. Threshold of toxicological concern, TTC. The threshold of toxicological concern, TTC, is a level of human intake or exposure that is considered to be of negligible risk, despite the absence of chemical-specific toxicity data. The TTC approach is a scientific rationale provided to estimate acceptable daily exposure values for compounds with limited or no toxicity information available. The approach was initially developed by the Food and Drug Administration, FDA, for packaging migrants, and used a single threshold value of 1.5 microgram per day, called the threshold of regulation. However, a more specific TTC approach for pharmaceutical manufacturing operations was developed by Dolan et al. According to the Dolan principle, there are three different categories of compounds on which the TTC principles can be applied in case limited or no toxicity data is available. 1. Compounds that are likely to be carcinogenic, ADE and PDE, are 1 microgram per day. 2. Compounds that are likely to be potent or highly toxic, ADE and PDE, are 10 microgram per day. 3. Compounds that are not likely to be potent, highly toxic or carcinogenic, ADE and PDE, are 100 microgram per day. For the first category, carcinogenic potential is assessed based on in vitro mutagenicity data and/or structural alerts for genotoxic potential and confirmed by an appropriate in vivo test. The second category contains compounds with limited data indicating they may produce pharmacologic or toxic effects at very low doses, compounds that show evidence of mutagenicity in vitro studies, but not confirmed in appropriate in vivo studies, or compounds with a positive in vitro study in combination with a negative in vivo study. The third class contains compounds with no a priori evidence of unusual toxicity or potency and which are not considered to be mutagenic, no structural alerts and negative in Ames test. When the TTC approach is applied, it is important for both risk assessors and risk managers to keep in mind that it is a probability-based screening tool and may have additional uncertainty. The TTC principle is based on oral acceptable daily intake levels but can be expanded to parenteral routes, i.e. intravenous, subcutaneous, intramuscular. Furthermore, the thresholds are based on chronic exposure, meaning that in case of an atypical event in cleaning validation an additional margin of safety is provided.